Greetings everyone, and today we are going to discuss about. So today we are going to discuss about plastids. First, we are going to discuss that who discovered plastid. It was discovered by Haeckel. It is a double membrane bound organelle. Double membrane bound organelle. All plastids are double membrane bound, as well as these plastids were named from the Greek word plastos, meaning form. Plastos meaning form. Why? Because they cause formation of several substances, several organic molecules. The chloroplast causing photosynthesis to form glucose, after which that is plastids are basically found in greater number in plants and protists. Which protists? The algae. After which we are having plastids. They are, they are involved, and first of all, these are derived from the early plastids. These are derived from the early plastids or proplastids. These are derived from specialized structures which are known as early plastids or proplastids, which are actually plastids in nature. But these plastids are immature and colorless plastids. Now, these colorless plastids are further differentiated into three types of plastid on the basis of their pigment, which on the basis of their pigment as well as on the basis of their structure as well as on the basis of their function. They are divided into the following types, which includes the first and foremost, that includes proplastic being divided into chloroplast, leucoplast, and chromoplast. Now, etioplast is a type of proplast. It is a type of plastid which is immature form of chloroplast, which means when there is no enough amount of light energy in order to convert the proplastid into chloroplast, the etioplast first converts the proplastid first converts into the stage of etioplast, and from the stage of etioplast it is then converted into the stage of chloroplast, which is the final working stage of chloroplast from where it can produce glucose through photosynthesis. After the production of glucose, and once there is um, the functioning of chloroplast, after the functioning of chloroplast, once the chloroplast once the chloroplast degrades its green pigment chlorophyll, it is then converted into chromoplast. As we know that chloroplast is green because of the chlorophyll as it reflects the green light. On the other hand side, when it shades, when it degrades its green color, its green pigment chlorophyll, it is then converted into chromoplast. As we have previously we might have discussed that what is a pigment. Pigment is any substance which is capable of absorbing a specific wavelength of radiation of light in the form of visible light. Pigment is any substance which is able to absorb any is a specific radiation of wavelength of light in the form of visible light, and that visible light is having its wavelength in between 380 nanometer to 760 nanometer. 380 to 760 nanometer. 380 to 760 nanometer. Now on the other hand side, leucoplast is an other proplastid which is colorless. We are gonna all discuss all of them, but for now we are gonna just discuss about proplastid generally. So these are divided into chloroplast, chromoplast, and leucoplast. Apart from this, when it comes to proplastid, we have two different types of cell in plants. First includes the meristematic cells. Second is the permanent cells. Now, when meristematic cells Meristematic cells are undifferentiated cells, which means these are dividing cells capable of cell division, and permanent cells are differentiated cells. Now, when undifferentiated cells, meristematic cells, are converted into permanent differentiated cells, then this process is known as differentiation. And when permanent cells or differentiated cells after differentiation are converted back into the undifferentiated cells, which means the gain of the capability of gaining cell division after losing cell division is known as what? Is known as de-differentiation. Is known as de-differentiation, in which the undifferentiated cells again convert into the differentiated cells, regaining their capability of doing cell division. After which, the cells which are obtained in result of de-differentiation are known as de-differentiated cells. After de-differentiated cells, when the de-differentiated cells are again converted into permanent cells, then this process is known as redifferentiation, in which de-differentiated cells are again converted into into differentiated cells. This process is known as redifferentiation. De-differentiated cells converted into differentiated cells. This process is known as redifferentiation. Or we can also say that the regain of the capability of losing cell division after regaining the capability of doing cell division is known as redifferentiation. Apart from this, redifferentiation and de-differentiation are two processes which occur during the secondary growth. During secondary growth in plant, the parenchyma cells are again converted into the meristematic cells, and then meristematic cells again convert into parenchyma cells during the secondary growth. So this process occurs in plants. Now, the meristematic cells contain proplastid. Meristematic cells contain proplastid. When meristematic cells are differentiated into permanent cells, then the proplastid are either converted into the chromoplast, or in the chloroplast, or in the leucoplast. If it is a green part of plant, then that would be chloroplast, because it contains a green pigment chlorophyll. On the other side, if there is the colored part of plant, such as the ripened fruit, as well as the flower's petals, then that would be having the chromoplast. After which, the roots of the plant will be having leucoplast, as these are colorless bodies again produced by the meristematic cells, or the process of differentiation. So the process of differentiation, apart from which, when meristematic cells are converted into permanent, this is known as differentiation. Now, on the other hand side, we are having one more feature of plastids, that they are capable of division. These are capable of division. These are capable of division. What does this mean? These rapidly divide themselves. As these rapidly divide themselves, these are having less metabolic activities. As these can rapidly divide themselves, these are having less metabolic activities, because as we know that during cell division, most of the material of cell is made inside the interface. Inside the interface, 90% of the cell is prepared for the cell division, while in the M phase, the actual cell division takes place. So we can say that the major metabolic activity takes place in the interface rather than M phase, because in M phase there is only the actual cell division, not the metabolic preparation or the metabolic activity for cell division, which is done in the interphase. Done in interphase. Apart from which, we are also having example of we are also having example of cotyledon. As you might have studied the structure of cotyledon, such kind of structure is present, and on the upper side there is the presence of plumule, which causes the growth of shoot, and on the downward side there is a presence of radicle, which causes the growth of roots. Now, plumule is having meristematic cells on its apical part. On its apical part or top part, it is having the meristematic cells. It is having meristematic cells, And same goes with reticle, that it causes the growth of roots, and that is having meristematic cells on its apical part. Now these meristematic cells again then convert into permanent cells, which are supposed to be either shoot or supposed to be either root, which is they are supposed to be either root or shoot, which contain plastid. If it is growing towards the root, then that would be leucoplast. If it is growing towards the shoot, then first it will be chloroplast, and after maturation, after the growth of the flower, then growth of the flower and ripened fruit, that would be considered to be as the chromoplast. Growth of the ripened fruit and flower, that would be the chromoplast, and before that there would be chloroplast. So this is a brief intro about plastids, after which we are having the chloroplast. What is chloroplast? It is plastid, of course double membrane bound because it is a plastid. So here we are having the structure of chloroplast, such a manner that it is double membrane bound organelle, largest organelle in the plant cell, after which it is having its inner membrane and outer membrane. Outer membrane is having protein channels, porins, as we studied in mitochondria. Same goes here. It is free, which allow free permeability of the material. Second inner membrane is selectively permeable, differentially permeable, and the space between inner and outer membrane is known as outer and inner membrane. Space between outer and the inner membrane is known as intermembranous space, apart from which this is having according to the endosymbiotic theory, it is derived from the photosynthetic bacteria. It was a photosynthetic bacteria some centuries or some era ago. So in that sense, that was also somewhat a photosynthetic bacteria which was engulfed by an early eukaryote, due to which it became the part of eukaryotic cell, according to endosymbiotic theory. Apart from which, it is having ribosomes, as you can see, it is mentioned in the diagram. It is having ribosomes, which is 70s. It is having the double-stranded circular DNA, extranuclear DNA, which is guanine cytosine rich DNA, which is known as plastidome. On the other hand side, there is the presence of enzymes, which is RNA polymerase, DNA polymerase, RNA polymerase 1, as well as Rubisco enzyme, ribulose biphosphate carboxylase oxygenase, after which there is the presence of several ribosomes, such as 70s ribosomes. There's presence of RNA, all three types of RNA, mRNA, rRNA, and tRNA. Apart from which, it also contains it contains several discs like structure. It contains um, let's zoom it a bit. So here, as you can see within the diagram, there are several disc-like structures. There are several disc-like structures over here. This is kind of disc-like structure which is known as a thylakoid, and that thylakoid is double membrane bounded. That is double membrane bounded, which is known as, and that double membrane is known as, that double membrane of thylakoid is known as the peristromium. It is known as peristromium. Double membrane is known as peristromium. Let me just write it over here. It's peristromium. Apart from which, when it comes to other material, there are also plastoglobules. What are these plastoglobules? These plastoglobules are material which are present inside these are actually globular proteins which are present inside the chloroplast for the stability of granum. Now here, these thylakoid are basically having what? These thylakoid are basically arranged in a stack. These are arranged in a stack up, just like a pile of books. So as these are arranged in a stack, and just like a pile of books, this complete stack is known as granum, and plural it would be called as grana. and one stack is made up of 50 or more than 50 thylakoids. One stack is made up of 50 or more than 50 thylakoids. One stack is made up of 50 or more than 50 thylakoids. Apart from which, there are 40 to 60 granum, there are 40 to 60 grana inside the chloroplast. Apart from which, each granum is connected with another granum by the help of a thylakoid, which is known as larger thylakoid, or stromal lamellae, or fret membrane, or intergrana. We call it either intergrana or stromal lamellae or fret membrane or larger thylakoid, which connects two grana together. On the other hand side, the grana are having thylakoid which are known as smaller thylakoid. These are generally known as thylakoid, but we can call them specifically as smaller thylakoid. Apart from which, we are having the lumen of thylakoid, the lumen of chloroplast. Inside the lumen of chloroplast, all of these materials are present, as well as apart from this, um, as you can see in the diagram, there is also the structure of also the structure of DNA, as well as the structure of RNA, ribosomes. Apart from which, we are having, all right. So apart from this diagram, the peristromium is in actual has it is peripheral to the stroma. It is peripheral to the stroma. One more thing, that this lumen of uh, chloroplast is having matrix fluid inside it. This fluid is known as stroma, and within the stroma there is the synthesis of several materials, which we are going to further discuss. So stroma, where there is the presence of the thylakoid membrane. There is a presence of thylakoid membrane in the form of granum, and the peristromium is peripheral to the stroma. Apart from which, overall, as we can write that it is a site of photosynthesis. As we have previously studied, it is a site of photosynthesis, and as it is a site of photosynthesis, we have also studied that inside the thylakoid there is light reaction or light dependent reaction, and inside the stroma there is light independent reaction or dark reaction. Both reaction take place during daytime. However, dark reaction can also take place during nighttime, but mostly both of these take place during daytime. Apart from which, we can say that here, um, this is a site of photosynthesis, and apart from this, we can say that inside this structure, these chloroplasts are self-replicative, as we previously discussed in the structure of mitochondria, that these are self-replicative. These can produce their own proteins. According to the endosymbiotic theory, they are derived from the prokaryotes, as well as these are the largest organelle in plant cell, as well as they peristromium. After which peristromium is the membrane of thylakoid, and apart from this, these thylakoid membrane, let me just elongate it and write it. Let me just draw the structure of thylakoid over here. So this is the structure of thylakoid. I'm just trying to draw a rough one here. As you can see, this is a double membrane which is peristromium, and within this double membrane we are having um, oops, within the double membrane there is a lumen of thylakoid membrane. Within the lumen, there are several pigments. Now, these pigments absorb specific wavelengths of radiations of a particular light, which are then utilized for gaining the energy from light energy and converting light energy into mechanical energy. Oh, sorry, it's from to convert light energy into food energy or chemical energy. Now, in that process, as it converts the light energy into the chemical energy, it is known as chemical synthesizer or food storing organelle. As we previously discussed, that plastids are containing pigments. So these, as these contain pigments, these are chemical synthesizer or chemical storage organelles. Now in this case, these are having several pigments. First pigment and is most common one is chlorophyll. Now chlorophyll is having four types: A, chlorophyll B, chlorophyll C, chlorophyll D. Chlorophyll A and B are found in plants, C and D are found in algae. On the other hand side, bacteria is also having chlorophyll, but it is not having chloroplast. It is bacteriochlorophyll. In bacteria, that is bacteriochlorophyll. On the other hand side, we are having carotenoids. Carotenoids are divided into two types. First includes the carotenes, while second type includes xanthophylls. Carotenes are basically having their color as red and orange. They give red and orange color to the plant's part. Xanthophylls give the yellow color to plant's part. Usually, as we can see that whenever we are having autumn season, the leaves of plant becomes yellowish in color and they start to fall down on earth. They become yellowish in color and start to fall down on earth because the chlorophyll present inside the chloroplast starts to remove and shade off, while during this process of removal of chlorophyll, the chloroplast is then converted into the chromoplast, and as it is converted into the chromoplast, the pigment which is left behind is xanthophyll, and that xanthophyll, being yellowish in color, provides yellowish color towards the leaves, due to which the leaves appears to be yellowish in color and orange or red in color when it comes to the autumn season, when plants grow in autumn season. So in autumn season, that occurs that the plants become yellowish, orange, red in color. That is why it is a condition that plants become yellowish in color and orange or reddish in color. Their leaves become yellowish, red or orange in color, sorry, and fall down on earth. That is because of the appearance of xanthophyll and carotene in the chromoplast. Apart from which, there are other pigments as well, such as lycopene, lycopene and lycopene is a pigment which reflects the purple wavelength. Sorry, lycopene is a pigment which reflects the red wavelength. Reflects the red wavelength. That is why it gives a red color to the plant. After which we have another pigment which is anthocyanin. Anthocyanin reflects purple wavelength, due to which it gives a purple color to plant. Now here anthocyanin is anthocyanin is providing purple color to the plant, and that is found in brinjal, as we are having color of brinjal as purple. Lycopene, red pigment, and cause it provides red color to tomato. That is why the scientific name of tomato is Lycopernisol pernisol estudicanol. Canol. So the tomato is having Lycopernisol estusenol, estusenol, senol, estusenol, something like that. I'm not sure about the spell, but it is the scientific name of tomato. That tomato is having its scientific name is Lycopersonol estusenol. Lycopersonal estusinol is the scientific name of tomato, and that is red because of the presence of lycopene, and that is why its name is also given as Lycopersonal estusinol. Apart from which, chloroplast is having its characteristic. Chloroplast is having its characteristic that it is able to move towards the sunlight. For suppose, let me just draw it with a diagram. So for suppose this is a cell. For suppose this is a cell, and this is a position of chloroplast. For suppose this is cell, this is a position of chloroplast, and this is sun. This is sun. Now, in order to gain light energy, the chloroplast is able to move towards the direction of sunlight by the streaming movement of cytoplasm cyclosis, due to which the chloroplast move towards the sunlight, the streaming movement of cytoplasm cyclosis, and faces the direction of sunlight in order to gain the light energy. This process is known as phototropism, in which the plant cells organelle changes its position with respect to light, changes its position in order to get into the direction of light. So that, let me just write it down. It is it is the movement of plant it is a movement of plant organelle it is a movement of plant organelle towards the it is a movement of plant organelle it is a movement of plant organelle towards the direction of sunlight due to internal stimuli. So in this case, it is a movement of plant organelle towards the direction of sunlight due to internal stimuli. And now here, internal stimuli serves as the need to gain light energy, due to which cyclosis occurs due to the endogenous activity of cell. Need of light energy, cyclosis occurs, chloroplast moves towards the direction of sunlight. On the other side, there is another term known as phototactic movement. Phototactic movement is a movement in which the plant's organ, not plant's organ, but plant's tissue or plant's organ moves towards the direction of sunlight. For instance, we are having shoot. Shoot grows towards the direction of sunlight, opposite to the direction of gravity. So it is known as phototactic movement. On the other hand side, sunflower faces the direction of sun. So it is phototactic movement. On the other hand side, there is example of roots. Roots do not face the direction of sun. Roots do not face the direction of sun. These are opposite to the direction of sun, and these are facing the direction of gravity, which means their growth is towards the gravity against the sunlight, due to which this is known as geotactic growth. Geotactic growth, or we can also call it as geotropism, because in this case the cells face the direction of gravity. cells face a direction of gravity, it is known as geotropism. And the growth of the root, geotectic movement. In that case, now coming to our main topic, that here we are having two phases. First, we are having here light reaction occurs, and as light reaction occurs, what will be formed? First of all, there will be the photolysis of water. Photolysis of water will occur, due to which H2O will be converted into two protons, two electrons, and two protons, two electrons, and 1 upon half oxygen. Now, two protons, two electrons, and 1 upon half oxygen is present. If you want to get two water molecules, if you are photolysing two water molecules, then they would be converted into four protons. Previously, there were two protons, two electrons, and now there will be four protons, four electrons, and one oxygen. So, as the process suggests, photolysis. Photo means light, lysis means splitting. So photolysis is the process in which the splitting of water takes place in the presence of light. In that case, water is being splitted by utilizing the energy of light, sunlight, due to which it is split into protons, electrons, and oxygen molecule, such that there are four proton, four electron, and one oxygen molecule during the photolysis of two water molecules. Now, in that case, this is a complete process we are going to discuss someday in a later video. But for now, this is the process in which the final product comes out as NADPH as well as ATP and oxygen, as you can see in the reaction. The oxygen which is firstly liberated here is of no use to the plant, so it is just given out of the plant cell through transpiration. So through the process of transpiration, it is given out of the plant body, after which there's ATP and NADP, which is utilized in the process of dark reaction. It is utilized in the process of dark reaction, and which is taking place in the stroma. And utilizing the energy of NADPH and ATP, there's a production of glucose by the fixation of carbon dioxide during dark reaction. We are going to further discuss that right now. That here, for suppose this is a cell, this is a cell here, not cell, let us suppose this is a chloroplast. This is chloroplast. Within that, this is granum, in which there's thylakoid membrane, of course. This is stroma. For suppose this is stroma, this is granum. Now, within this, first of all, water molecules come inside the granum. Water comes inside the granum. Now, inside the thylakoid, photolysis occurs, due to which there is the production of ATP, NADPH, as well as oxygen, which is liberated after light reaction. On the other side, some of the oxygen is liberated for transpiration, while some of the oxygen is utilized in the process of dark reaction. Is utilized in the process of dark reaction. Is utilized in the process of dark reaction. So during dark reaction, what will happen? There will be the fixation of carbon dioxide, such that carbon dioxide will be fixated using the enzyme Rubisco by fixating carbon dioxide with ribulose biphosphate using the enzyme Rubisco. Ribulose biphosphate carboxylase oxygenase, which causes the fixation of carbon dioxide into phosphoglycolate 2 carbon sugar, after which further cycle goes on in a cyclic manner, due to which the final product obtained is CH2O. This is an empirical formula of carbohydrate, not getting much time here. So CH2O here, which is the empirical formula of carbohydrate, which means glucose is produced. Now, ribulose biphosphate causes the fixation of carbon dioxide, apart from which uh, ribulose biphosphate carboxylase oxygenase. First of all, it causes Here, ribulose biphosphate carboxylase is having two functions. First includes the fixation of carbon dioxide, which is known as carboxylation. Second includes the removal of oxygen, which is known as oops, rubisco. First of all, it's rubisco here. So rubisco, it is having two functions. First includes the fixation of carbon dioxide, which is carboxylation, and second includes the addition of oxygen, which is known as oxygenation. That is why it is having the capability of binding with both oxygen as well as carbon dioxide, due to which it is able to then fixate carbon dioxide, carboxylation, and add oxygen, oxygenation. Apart from this, when oxygen is received here, of course, the process of fixation of carbon dioxide occurs, glucose is formed using the energy which was gained during the light reaction in the form of ATP and NADPH. That is again converted into ADP, NADP plus, because it is losing its proton, due to which it becomes plus. First of all, uh, it was in the reduced form. Now it is not in the reduced form, oxidized form, so it becomes plus NADP plus ATP, NADP plus, as well as glucose, which is written above. So this is what we gain during this process. Hopefully you understood what is about today's video. Hopefully we'll meet again in